अच्छा अच्छा, 15 किलोमीटर इज नॉट फार एट ऑल.
हाँ,
बहुत नजदीक है फिर तो. आपकी रोड बहुत अच्छी है.
पूरे गुजरात में आफ्टर 2002 when Narendra Modi became chief minister, he has changed the entire.
Yes, no doubt, no doubt.
We are
बहुत
live
सारा
now.
सारा परिवर्तन इंडिया में भी हो रहा है उनके नेतृत्व में.
So shall I present your screen on behalf of you, or you will
Yeah,
be
you,
able to?
I would request you to do it, please.
Yes, sir. Yes, sir. Sure, sir.
And I'll request you to next slide, then you can change the slide.
Yes, sir. Yes, sir. Absolutely.
कितने बच्चे हैं आपके यहाँ ध्वनि जी?
More than 30,000 बच्चे हैं. 30,000.
Oh, that's a big.
We have more than 34 institution. We have a 200 acre of this land where about 100 buildings are standing, and we have hostel capacity of about 12,000.
Oh my God!
It's bigger than one town, where there are all facility. We have about more than 20 ordinary food court and 12 branded food court where any food is available. And the hike of this is that we have about more than 1,500 foreign nation students from 56 countries.
Vijay sir, shall we start?
It's left on you. You can start.
So it is rightly said, people who succeed have momentum. The more they succeed, the more they want to succeed, and more they find a way of succeed. Success is not just about you accomplish in your life, it's about what you inspire others to do. Today's esteemed guest speaker is one of such personality who is an inspiration to millions of people around the globe, whose entire life is an experimental learning, motivation, and encouragement for the world. A very pleasant morning to everyone. I, Dr. Dhwani Upadhyay, Assistant Professor, Microbiology Department, with a delightful smile on behalf of Parul University, feeling honored to welcome our today's embryo speaker, renowned Indian scientist, distinguished professor at Indian Institute of Technology, Padma Shri, Dr. Syed Ahtam Hasnain, sir. We are truly honored to have you with us, sir, today. I am delighted to welcome our Honorable Dr. Emin Patel, sir, Provost, Parul University. I would like to also welcome our Dean, Professor Trilok Akhani, sir. I also welcome all the faculties and all the young ignited minds. My dear students, to this one of the kind of live webinar, I assure you all that experiencing this live webinar will give you life learning lessons and boost you to achieve your goals even greater zeal and passion. I would like to request Honorable Provost Sir, Dr. Emin Patel Sir, to say a few words on this August occasion. Sir, please.
Ji, namaskar to one and all, and very good morning. No doubt, as Sir has asked about what is the Parul and how and what are the numbers. So we have as 34 institution, and all in institution have a this or that program going on. So this is today's my second program where I have just connect, and especially the stalwarts we have got Professor Syed, Padma Shri. So obviously it is my duty to connect in this program. So sir, I will welcome on behalf of the Parul University, being Vice Chancellor of the university. I consider our privilege to be people like you, those who are the contributor in the area of biological science, as we know that the science is the area where there are a lot of research going on, and people like you, who have a very good experience and can give some of the tips to our teachers and students and participants. Obviously, they are maybe very much beneficial to that. As so far this program is concerned, I also appreciate Dr. Dhwani to just have invite the people like you, because it's now become easy for us in this digital era to invite anyone from any corner of the globe, because to travel and everything is where sometimes limitations. But as we are in the digital era, and this corona has taught us so many lessons, and one of the lessons is that everything is a digital and in virtual form. So sometimes it became very easy for anyone to connect, and obviously that's why we are lucky enough that you are people like you, those who are a high demand people will give the contribution to that. And as you said that you have delivered the talk on MS University. MS University is a state university. while this Parul University is a private state university which was established in 2015. But we have grown since last 6 years like anything. As I said, that we have a 200 acre of campus where we have 34 institutions, 100 buildings, 12,000 students are staying in the campus. That capacity is there, but presently due to this corona, hostels are almost vacant because no one is staying in the campus. But we have a 5 hospital. Out of that one, due to this MBBS program, we have a 650 bed hospital in which we have just give the social service to the entire area. As so far COVID hospital is considered, it was considered to be one of the best COVID hospital, and we have treated so many patients in our hospital. We have our own oxygen plant we have established before March, so there was no problem. And like that way, we are giving service to the society by various ways. We have a social responsive cell by which we are just guiding the people around this Vagodia district, where all sorts of help, either in terms of advice, in terms of medicine, in terms of social service, we are providing. So like that way, Parul University, we have logo that be here, be vibrant, and we are really the vibrant. And as far as the activity is concerned, you can see that we are doing a lot of things, and by which our goal is to prepare our student not only for the degree, but to all-round development of the student, by which we have created about more than 30 cells, where more than 100 dedicated mentors are working. So wherever student is interested to nurture his ability or talent, we are providing the platform. Along with that study, they are also developing their own talent in various areas, and all the cells are functioning with separate dedicated mentors, and that became the attraction of the student. And that's why we are getting a very good size of admission across not the any state of India, but across the globe we are getting the students. So like that way, we are lucky enough that here there is a lot of diversity as so far cultural exchange is concerned, as so far variety of student is concerned, and we are happy that we are growing very, very fast. And it will be added when people like you will give the talks. Obviously, it will be very much pleasure for us. So I welcome you again, sir, and hope so that in future also, as and when required, if it physically possible, when also we will invite to you if your time permits. And my best wishes to all participants that you are lucky enough that we have a stalwart speaker, resource person who is going to talk with you. And I also request all of you, if possible, then chat box, you can put some question so that our expert can give the answer of it and some doubts of clarity to you. So best wishes to all of you, especially the PIES. I think Principal Trilok may not be present, but I hope so that you all are working very well. We are expanding as so far science wing is concerned. We are getting about 800 admission every year in the science wing. So obviously our science is going to be strong with all physics, chemistry, maths, and all branches. So like that way, we are growing, we are developing, and as so far this type of more and more program will definitely become the attraction of students to study in the Parul University. So with this word, my best wishes to all of you. Thank you. Over to you, Dr. Dhoni.
Thank you. Thank you. Thank you so much, sir, for your enlightening words. Bacteria don't stop at frontiers, and neither should scientists. Professor Hasneen, sir, is a creator and originator himself in an area of bioscientific research for decades. Though Honorable Sir does not need any introduction, as he himself is an epitome of inspiration and golden feather of our nation. I would like to take this opportunity, and I feel privileged to introduce our emeritus speaker, Professor Dr. Hasneen Syed, sir. Professor Sir Syed Hasneen has completed his bachelor's and master's from Magad University. He opted for MPhil and PhD from Jawaharlal Nehru University. Sir Padma Shri is recipient of Germany's highest civilian award, the Order of Merit from President of Germany. The Padma Shri award is provided to Sir by the President of India. This is a very important award which is given to the very important persons only throughout the world, throughout the India. He is internationally accomplished for his work on DNA fingerprinting and tuberculosis. Based on his work on Indian basmati rice DNA fingerprinting, India won the case against Pakistan, enabling continued export of basmati to UK, preventing a major economic loss to India. Subsequently, Ministry of Commerce, Government of India established CDFD APEDA Basmati DNA Analysis Center, where all Basmati exports are certified. Professor Sir is an institutional builder policymaker, and a scientist. Recipient of PhD from JNU Delhi, DSc, DMed as he taught at Delhi University as a lecturer and left for Canada, USA for postdoctoral fellowship. He joined National Institute of Immunology as first director, Center for DNA Fingerprinting and Diagnostic, a DBT institute, and within 6 years, CDFD became a gold standard for forensic crime investigation, and all high-profile political cases were referred to CDFD by High Court or Supreme Court of India. CDFD started attracting several Nobel laureates as a Foundation Day lecturers. The international meeting organized by him in Hyderabad on new biology and justice delivery system, inaugurated by President of India, attended by Supreme Court, High Court judges, laid the foundation for DNA profiling bill, which he drafted. He was invented as a Vice Chancellor, University of Hyderabad, and during his tenure, University of Hyderabad was firmly established as first University of India, displacing JNU or Delhi University. The National Assessment and Accreditation Council, that is NAC, awarded University of Hyderabad highest score ever given the International Scopus Databases Analysis, ranked UHO as the top Indian university. This was all possible because of Sars' contribution. Sars joined IIT Delhi as a professor by invitation and served until superannuation. Under his leadership as Vice Chancellor, Jamia Hamdard climbed fourth year into higher orbits of excellence and was recommended by MHRD as Institution of Eminence. The fifth-ranked private-funded institution functioned in India. Professor Sir is a recipient of many prestigious awards such as ICMR's B.R. Ambedkar Award, the highest award for excellence in biomedical research, G.D. Birla Award, Shanti Swarup Bhattacharya Prize, considered as a Nobel Prize and given by the Prime Minister, FICC Award, which is given by the President of India, J.C. Bose National Fellow Award, Renaxby Research Award, Goyal Award, Masson Award, etc. International recognitions include the rare Humboldt Research Prize, the very exclusive Robert Koch Fellowship of the Robert Koch Institute, Burning. The third Indian prize or honorary doctorate from Queen's University is provided to Sir. Hasnain Sir was a member of Science Advisory Council to the Prime Minister and was a member of Scientific Advisory Committee to Union Cabinet. He was Chairman, Biotech Advisory Council, Government of Andhra Pradesh. Sir is an elected fellow of the prestigious American Academy of Microbiology, USA, and all major science academies of India. He is the first Indian elected as a member of German National Academy of Science, Lapodonia, and youngest to be elected as a fellow of TWAS. He was a member of International Advisory Board, Europathogenomics Network of Excellence. He was a member of Science and Engineering Research Board, Ministry of Science and Technology, Governing Council ICMR, Ministry of Health, Research Advisory Committee of AIIMS New Delhi. He was the Chairman Academic Committee of PGIMER Chandigarh and member of Institute Body and Governing Council of PGIMER, member of Review Committees of TIFR Mumbai and TISS Mumbai. He served as member, University Grant Commission, Governing Council of ILBS, Vasant Kunj, Delhi. Indian Institute of Advanced Studies, Shimla, IISc Bangalore, IIM Kozhikode, IISER Bhopal, JNU New Delhi, AMU Aligarh, and several other institutions, policymaker, bodies of Minister of Science and Technology, MHRD, Healthcare Defense. He continues as a member of Agna Khan Foundation, National Committee, Board of Director, Audit Committee, Remuneration Committee of several companies, Executive Council, etc. He has selected as ACRB National Science Chair at Department of Biochemical Engineering and Biotechnology at IIT Delhi with more than 300 publications in high impact journals and dozens of patents. He has mentored approximately 150 PhD, MD, postdoctoral students, four of whom received the prestigious INSA Young Scientist Medal. Sir leads a very large, very well-funded and active group of researchers in the area of tuberculosis and latently in COVID-19. It's my privilege to invite Sir to deliver his talk and enlighten us with his knowledge and lifelong experiences. Sir, we would be honored to hear about your research experiments and experiences. Most welcome, sir.
Thanks a lot, Parulji. Can you hear me?
Yes, sir. Yes, sir. You are audible.
Very good morning to Professor Patel, the Vice Chancellor of Parul University. It's a pleasure to be at Parul, although I'm physically not at Parul, but I can tell you I'm mentally and otherwise emotionally with Parul, more so because I see Parul will one day, the rate at which you are growing, in just about 6 years, you have achieved this level of excellence. I will not be surprised if in another 5-10 years you become one of the top universities of this in this part of the world and one of the top place in the world. Congratulations, you have a very vibrant faculty, you have a very vibrant students, a large number of students, and the social work that you are doing. These will also reward you with 5 hospitals, with COVID treatment, free treatment. I think these are things which will really take you very, very far. And I'm very happy. My only regret is, हमारी जो इंट्रोड्यूस किया जिन्होंने मुझे, उन्होंने 15 मिनट मेरा ले लिया मुझे मेरे बारे में बताने के लिए. आज के डेट में इसकी कोई जरूरत नहीं है. You just give the website, do a Google search, and everything you will know about the person. But thank you so much, Dhoni ji. Thank you so much for your kind invitation. When your invitation came, I thought it's a private university, and coming from a young faculty member, I must accept. And that's something, I've been really fortunate to get a large number of people attracted to my group. And I thought it will be a good opportunity to get also students from Parul University to join us in our quest for knowledge. She asked me to give a talk as a public talk on the area of common interest. And I thought today, what can be more important than the COVID virus that causes COVID-19, the Sars Coronavirus 2. And that's an example of how biological sciences and medical sciences are today making life such more difficult in the terms of new discoveries and new pathogens coming up. And that's where this science will continue to thrive. And I chose to talk today on research in biological sciences will dominate all sciences in this century and beyond. Thank you once again. If I can look at the next slide. The next slide will tell you about how things have been working in the age of engineering. Just a century ago, it was the age of engineering, and examples are for all of you to see, whether it's a locomotive, whether it's the slide movie projector or the cycle, or even the Eiffel Tower. These are all examples of excellence in engineering sciences. Then came, next slide, please. Then came the age of, next slide. Then came the age of chemistry and physics. And we now know many advantages that we see today, a routine thing, including X-ray discovered by Röntgen. And I happened to visit his place where he worked. It's been converted into a museum now, the first X-ray machine. And many other things today that we take it for granted are actually discoveries made because of the contributions of people working in the area of chemistry and physics. So 20th century, a century later after engineering sciences, it has become very, very common contributions of chemistry and physics have benefited humanity like never before. Today, while we are in the 21st century, next slide, please. While we are in the 21st century, it is clearly considered as the age of biology. And in the next slide, I'll tell you the long distance the biology has covered in these many years. Ever since the discovery of the microscope by Liu Van Hoff, and where he described a single cell organism, to today, many years, many decades, many centuries later, we are talking about the Sars Coronavirus and the disease, the COVID-19 that it causes. Now, Sars Coronavirus, COVID-19 is not a discovery which has been made by scientists, but it's a discovery that was forced to be discovered because of the problems this virus has been creating and affecting the world. So it's a pandemic, and in the process, we have actually looked at, crossed many other milestones, including the most phenomenal part of the work, that is the 1953 discovery by Jim Watson, Francis Crick, and of course, two other people, of which the three of them got the Nobel Prize for the structure of solving the structure of the DNA. And later on, of course, many other developments took place, the recombinant DNA technology. And today, we are literally trying to figure out, where are we? Can we really conquer this disease, COVID-19? We thought we are the smartest creation of humanity. And today, when we look at the Sars coronavirus, we find this virus is perhaps far more intelligent than humanity, intelligent than human beings. When I made this statement in one of the lecture, one of the students asked me that, sir, you are saying SARS coronavirus is far more intelligent. So that means, does the coronavirus has a brain? That was a very interesting question. The virus does not have a brain, but it knows how to manipulate things in the human body without actually having a brain, making us biologists wondering, what are we really looking at? And if I can move to the next slide, which now then tells me, next slide, please, which now then tells us about the breakthroughs that have come in biology, which are not just from biologists, but from non-biologists, people who have never studied biology. It is they, when they moved to study biology, made fantastic contributions, path-breaking contributions. Here are some of the examples that you can see. Of course, the very classic photo of Crick displaying the double helical model, and Watson, who is still alive, looking at the model and seeing how this model could be. And Rosalind Franklin, who actually played a very major role in developing in doing the X-ray structure of the DNA crystal. Unfortunately, she couldn't live long enough, otherwise she would have also gotten the Nobel Prize. And she obviously could not. There are many other examples of non-physics, non-biology people, including our own Indian person who actually received his degree from MS University Baroda, Venkatraman Ramakrishnan, we call him as Venki, a very down-to-earth person, a physicist by training, who got the Nobel Prize for his discovery for his discovery of the protein translation machinery and the ribosomes that are involved in it. Next slide, please. So Nobel Prizes in physics and chemistry, if you look at the last 10 years, you'll be surprised to know that Nobel Prizes are also given in the area of medicine. But in the area of physics and chemistry, the Nobelists who win, they win the prize because of their contribution to biology. So, biologists have two more areas where they can compete for and get Nobel Prize, not just only in physiology and medicine, because the advances made in this are also all affecting humanity and human well-being. So what has been the impact of this DNA discovery and the revolution that we know we can see today happening? The impact of the gene revolution, we call it. Several things, including the very well-known now today DNA profiling. In fact, DNA profiling has become very, very common. Almost every place. And India, we were the first to set up the DNA fingerprinting at the Center for DNA Fingerprinting and Diagnostics in Hyderabad, where I served as the first director. And then, of course, now all over India and many parts of the world, DNA it's a best answer to solving crime, because trucks can, Volkswagen can become a truck, something can change, evidence can disappear, but DNA cannot change and DNA cannot lie. And that is the reason why evidence based on DNA have resulted in conviction in a very large number of, and almost 100% of the cases based on DNA. Then there are also new diagnostics and predictive medicine. I'll give you examples from work that was done by us when I was working at Hyderabad. And in fact, today I am in Hyderabad. In fact, I talked to Dhwani yesterday. Can you postpone my lecture so that I can give my talk when I'm back in Delhi? She said, "नहीं sir, वो नहीं हो सकता." हमने कहा, "आप मालिक हो, नहीं हो सकता तो कोई बात नहीं. I'll do it here only." So I'm in a meeting at a place in CCMB. I've asked them to please excuse me for about 1 hour, 1.5 hours, and they have been very kind to give me this room. Of course, I have my own laptop. Then cloning of humans, cloning of genes is very well known, but cloning of humans, stem cells, genetic modifications and genetically modified organisms, and of course synthetic biology. That means making life in the test tube, and genome editing. That means you can change the DNA that you inherited from your father and mother. These are all advancements that are today possible because of advancements in new biology. Next slide, please. So we can go to the next slide. DNA profiling. Before that, yeah, this one, right. DNA is a tool, as a tool to determine identity. That is what it is. And that's where it found its applications in crime solving. And this work was done purely as a simple experiment done where Jeffries was trying to do southern blotting. And when he used the DNA from one individual and using his probe, the band position shifted when he used other DNA. And he was wondering, "Aisa kyun ho raha hai? Why is the band position shifted when I'm using that? Although I know it's the same thing." Then it turned out to me that you are actually looking at sequences which are hypervariable. And that is what led to the first series of DNA fingerprinting bands, DNA fingerprinting probes. And after that, it's all history. DNA fingerprinting has become an established tool, a technology, not only in crime solving, but it's also shaken the foundations of humanity. We all live and grow with the fact that mother is a fact. I know my mother because somebody saw my mother delivering me in a hospital or a nursing home or a midwife in the home or someplace. But I don't know who my father is. But I believe that X is my father because my mother self tells me that he is your father. So mother is a fact, no questions about it. Father is a belief. And this is a notion in which the society has been living all these many years, ever since human lives came into existence. But today, even mothership, whether who is my real mother, is contestable, is questionable. In the CDFD, first time we found a case that came to us where two mothers were claiming to be mother for a single child. At that time, it was not possible to test that. We can still test it and prove that no, you are the lady X is only the mother of the child and not lady Y. But you know what today you can do? Today it is possible for two mothers to biologically contribute their DNA to a single child. So you look at how science has evolved. Look at how information has grown. Look at how technology has grown. So that's where I think it's shaken the basic foundations of our society. Not only we are now able to resolve who is our father based on DNA technology, but the belief that mother is a fact, even that belief is now questionable. You can, in theory, have a child born by contributions made by two independent mothers. So who would be called the real mother? These are things that society will now have to worry about it. Next slide, please. Of course, we worked together to create the DNA profiling bill, and I'm very happy that I was assisting the parliamentary committee in addressing some of the key questions that the Rajya Sabha had about the bill, and now hopefully the bill would be passed by the parliament this time. Predictive medicine. What do you say predictive medicine? Today, when you go to the doctor asks you some questions and then gives you some tests to do. What are these tests? These tests are there only to rule out disease A, disease B, disease C, disease D, and then say, I can't rule out disease E, so you must be down with disease E. So you are actually doing disease diagnosis by elimination. Today, it's possible to do disease diagnosis by not elimination, but actually saying so. When I was working at the CDFD in Hyderabad, we collaborated with the LV Prasad Eye Institute in Hyderabad, which is one of the best eye care center in this part of the world. And there we were looking at a disease which is known as primary congenital glaucoma. What is primary congenital glaucoma? It's a kind of a blindness, kind of a disease that makes you totally blind. And the worst part is, no child is born blind. Very rarely, not with this disease. But as the child grows old, and when he or she becomes 12 years of age or 13 years of age, he becomes completely blind. So there's a progressive decline in your eyesight so that you become completely blind. Imagine the plight of a father and mother when they find out that the child was born absolutely normal and जैसे जैसे बच्चा बड़ा हो रहा है, ये तो बिल्कुल he can't even see. And that is what is the disease known as primary congenital glaucoma. And we decided to find out, and in Andhra Pradesh, because of a lot of inbreeding means marrying within the family, this disease is very rampant, very, very rampant. So we found out that it's a genetic disease, and we found out a simple way. On your left, the black band that you can see in which on the black band, you can see white strips. If you don't see that white, that means this child is having this disease. Similarly, in the extreme right panel that you see, there's a last lane that you see, there's a white band is missing. That means this child is having this disease. So what if you can diagnose that this child has a disease? Then you can take this child to an eye surgeon. He will do a small operation and correct the problem. So the child, the sooner you diagnose it, the sooner the child will stop getting into this disease. He will lead an absolutely normal vision. So this has now completely changed the scenario. Thousands of children born in Andhra Pradesh and Telangana have now benefited from this work that we did at Hyderabad. It's already published and it's already in common practice. So this is where I think we can predict the child and say, you are going to have this disease. We want you to come and see a doctor and get operation done. Otherwise, you'll become a blind. So this is where disease diagnosis is no longer done by elimination, but by affirmation, a positive affirmation. Next slide, please. And now we have this challenge, the next generation sequencing challenge. What is next generation sequencing? You know, Sanger and Maxim and Gilbert, these two people discovered the DNA method for DNA sequencing. Sanger is one of the very rare person to get Nobel Prize twice. One for sequencing protein, one was for sequencing DNA. Now it is much more easy to sequence. The human genome sequencing cost a few $1 billion and about a decade to sequence the first human genome. Whereas today, I can get my DNA tested and sequenced completely and given in a small pen drive in a matter of few hours. And then people can look at my profile and say, oh my God, you are likely to come down with this disease. A very famous actress in the US, I will not name her, she got her DNA tested and it turned out to be that she has a very high chance of getting breast cancer. And this lady did not want to go through the breast cancer treatment and all that or a disease. She got her breast removed. This is where, you know, you can go to the extreme where you can actually do a diagnosis and then based on the diagnosis, treat yourself before the disease sets in. So that is possible. The question now then is, today, United Kingdom, the UK, England has started a project, 100,000 genomes. In India, we have also started a similar project to sequence the genome. But the question is, if I pick up individual A, B, C, D, E, which individual genome will I say is the healthiest human being? I may appear to be very healthy today, but tomorrow I can come down with the disease. So will my genome be considered as the healthiest genome? as a wild type genome? Or what is the basis? Now, that difference is because between my DNA and let's say the DNA of anybody in the audience today, or Dr. Dhwani Upadhyay's DNA, the difference would be hardly very 0.0001%. And yet we are so very different. Next slide, please. And the reason, and that is what is known as single nucleotide polymorphism. The difference between a chimpanzee and a human is only 5%, less than 5%. So tomorrow, कोई किसी ने आपको अगर कह दिया तुम बिल्कुल बंदर हो, तो वो गलत नहीं है, वो 5% ही गलत है. He is only wrong by 5%. 95% chance are that he is absolutely correct, because बंदर का DNA और हमारे DNA में अंतर मात्र 5% का है. And less than 1% DNA makes, look at the example that we have given here, one president of US, other president of US. Look at these two people. There are two people, are contrasting people. No comparison between the two. Yet they only differ by less than 1% of DNA. So difference in genome, even as less than 1% or 0.0001%, can make a big difference. And that is where a small difference in the progenome can have a real big impact. Next slide, please. And it now brings me to the concept of cloning. It was a wonderful work done by colleagues in the UK, where they cloned a sheep and they called the sheep as Dolly. And this sheep was known as a clone of the mother. And what they did, they took the mother's DNA and without doing fertilization with the father's sperm, they created a new offspring. They named it as Dolly. Of course, Dolly creation took a very large number of attempts. It was not possible in one attempt. It had hundreds of attempts were made. Then only Dolly could be created. And the worst part was Dolly could not live for long. And that is one of the challenge of cloning. The clones do not live for long because there is something which is coming from outside. It could be, we still don't know, which could be responsible for that. So Dolly had to be put to sleep. It was injected with a dose of that will cause death, because she was going through so much of pain and problems. So she did not complete her full life. She started getting old much earlier. So, but the fact is, today it is possible to create and clone an animal. And today, if I have a pet, I have a khargosh, I have a cat, or I have a dog, and I'm so attached to the dog. I used to have a dog, a German Shepherd. It stayed with me from birth till 13 years that he lived. And we got so attached to him that when he died, I felt as if a member of my family has left me. And that is where I decided that I will not like to have any more pets. But if I had been living in America, I would have taken my dog Rufus, who the name was Rufus, to the vet clinic, given a small saliva of Rufus, and in a matter of few weeks or a couple of weeks, I would have gotten a clone of Rufus, who will be with me for the next 13 years. Then later again another clone. So my Rufus, my pet, will never leave me at all. So it is possible today to clone your pets perpetually. It will never die. Before it dies, you make a clone, it's back again. Whether it's human being, it's possible or not. When I was working at the CDFD, a request came to me from somebody in Latin America saying that they have made a human clone and they would like us to test if it is indeed a human clone. We wanted more details. Details never came and we never could do the project. It could have been a hoax. But the fact is, it's a challenge to make a human clone today. But I don't know, somebody can beat the challenge tomorrow, day after tomorrow maybe, and it's possible then, it may be possible then to even make a human clone. But forget about the human, at least animal clone is possible immediately today. As I said, there are clinics in the US and Europe where you can take your pet and it will give you a clone of your pet in a matter of few days. And then that pet will be with you. You don't have to worry about the pet dying. So cloning has become a reality. Next slide, please. So that's where we are moving. Stem cell. Why stem cell becomes so popular? You know, when the human creation is made, when human beings are born, you know how they are born? a single sperm goes and fertilizes a single equivalent molecule cell of the female, the ovum. A single sperm and ovum merge together, fuse together to give a fertilized single cell. That single fertilized cell, as a function of 9 months, give rise to a human body. A human being is brought, comes into existence. The question is, if a single cell can become later on in over course of 9 months, a brain cell, an eye cell, a nose cell, a tongue cell, lung cell, heart cell, which means a cell is in principle capable of getting metamorphosed, getting transformed into a different set of cells and functions. And that is what gave rise to this inquiry of stem cell. They are the stems from where others will come. And today, Chinese researchers have been able to clone the tiny mouse from a skin cell. So using the skin cell, clone a mouse. Today, it's possible to grow fully functional teeth in mice from a single stem cell. And this success is going on. So stem cell therapy is going to be a reality, is already a reality. In places in Delhi, and I'm sure in Gujarat and Mumbai also, and many other places, there are stem cell depositories. I want to make sure that tomorrow, if I need to change my let's say my have problem with my heart, I want to change it. I want to get a heart replacement. I will not find anybody. So let me give my stem cell so that somebody can keep a heart ready for me tomorrow. And it's actually stem cell banks are created and are functioning and delivering. So that is where regenerative medicine has come in a real big way. Next slide, please. So that's a success of adult stem cells. You know, I have not gone to Gujarat during the Makkah time, you know, the corn time. But in Delhi today, it's a corn time because a lot of this is monsoon time, and we get a lot of makai, bhutta we call it, or whatever I don't know what you call in Gujarat, which you roast it on a charcoal and eat it. But when you go to a shopping mall or something, there also you get corn which is not roasted, but it actually boiled corn, big size corn, very sweet corn. You know what that corn is? That's a typical American corn. And those corns, I'm told that all of them are genetically modified corn. Genetically modified for two things. They can be very sweet and their size is bigger, and also they will last longer. So today, everybody is consuming it. So it's a reality today, genetically modified corns, food, and other things. So today, it's also possible to create, or as it is, resistant crops, cotton. I think Gujarat is one state which has the largest population of cotton, resistant cotton being populated in the country. What do? They produce a toxin which is produced only in the leaves. We don't need the cotton leaves, we need the cotton fiber. So fiber mein wo toxin nahi jati, leaves mein jati hai. So when an insect feeds on the leaf, it dies and it will stop feeding. And that's how it's known as insect resistant cotton. And similarly, genetic modified potato is coming, modified soybean is coming, canola is coming, and many others. In India, we don't have so many, but in India, it's still considered to be the world's second largest producer of genetically modified crops. So I think it's coming, it's in the horizon tomorrow. The golden rice that people are talking about, the corn, already we know, the soya, the canola, and of course, many with increased starch and increased oil. The flavor saver tomato in the US, it's being sold all over. You can make out that this tomato lives longer, and it's bright, very deep red, and it's nicely tasting. That's a genetically modified tomato. So we know that these things are safe. Of course, there are some groups that still believe that they are not safe. I think it's a problem with us. We are not able to communicate to them properly. Next slide, please. So that's where technology is going. And we now move towards one of the very interesting development, the synthetic biology, creating life in a test tube. It is possible today to create a new organism completely synthetically. And the best example, the only example which became a big time hit was the polio virus. Polio, as we all know, has been removed from the world, in most cases. So, and this has been done by looking at what is the genome of the polio. Get this genome synthesized in a chemical company, put it together in a laboratory, and make a live polio virus. This paper created a lot of interest, lot of sensation, because people thought if today you can create polio, tomorrow you can create more dangerous viruses. And one of the controversy which is going around in the world, including in the WHO, is about coronavirus. Is this a genetically created virus, or at least a genetically modified virus, or is it a naturally evolved virus? We still don't have answers to that, but then it is possible. So synthetic biology allows you to create life in a test tube. Next slide, please. There is another angle to it. We all believe, and that's what I said, biology today has affected social dogmas and social beliefs like never before. We all know that we are what I am because of the 50% of my DNA having come from my father and 50% of the DNA having come from my mother. Today we know that is absolutely true. But if that DNA together makes 100%, 10 times more DNA comes from bacteria that live in our body, or processes that have nothing to do with the DNA. It's to do with the way DNA is modified, or epigenetics, not genetics, but epigenetics. So epigenetics has been involved, has been known to be associated with a large number of disease, the heart disease, the obesity, autism, many diseases. Identical twins acquire discordant epigenomes during the life course. You would expect identical twins to behave identical. They don't. Why? If the DNA is 100% identical, because the epigenetic modification may be very different depending on what kind of epigenome they have. And that's where it comes, epigenetics becomes a very important. Next slide, please. And today it's possible to do genetic manipulation without disturbing the DNA completely. Let the DNA be there. Only I'll change the gene which is a defective gene. And that was done in taking example of E. coli. And then, of course, today it is possible to do even gene editing in humans. So that before the child is born, if I know that child carries this disease, and that can be done by gene prediction, then I can change that gene, and then so that the child will not have that gene at all and not have that disease at all. And for this kind of a gene editing work, a German scientist, along with two other scientists, got the Nobel Prize in 2020. And this is a very well known today CRISPR-Cas9 system. Of course, there are many other systems now to do genome editing. Next slide. I'm not going to go into details of this. I was talking to you about the bacteria that live in our body. So these bacteria are also known to be responsible for a lot of other things. And they are not small number. They are 10 times the DNA that exists in our body. And these bacteria are now known to be associated with diseases such as diabetes, asthma, rheumatoid arthritis, obesity, cancer, you name it. Almost every disease can now be associated with the kind of bacteria that live in your body. You know, one simple example, you live in Gujarat. I'm today living in Delhi. Earlier, I was living in Andhra Pradesh for about 13, 14 years. When you move from one city to another city and stay there for 4 or 5 days, the first thing that happens to you is that your stomach becomes upset. Have you wondered why the stomach becomes upset? Because the local flora of bacteria that's existing in your body, which is partly coming from the food that you have been taking, is facing a new kind of bacteria because of the new kind of food that you are taking. Of course, last 1.5 year, I have not gone abroad at all because of the COVID. But otherwise, I used to go quite frequently, travel abroad. And I used to have this problem quite often. Every time I change my food habit, my gastric system will be disturbed. The reason is your microbiome, the microbes that live in your body are trying to adapt to the new food through which these microbes come. And that is where, don't be surprised if in the next few years, if you go to the doctor will tell you, आप आज ये bacteria का culture है, take this culture of bacteria as your drug. No need for any drug. So tomorrow's drug would be a glass of a bacterial culture. That is where biology is heading. That is where science is moving. Next slide, please. So this is a more of a nutshell of where health is impacted because of the developments in technology and new biology. Today, there's a system, known as tricorder. It's based on a Star Trek, the serial. It works with your iPhone. All you have to do is to simply on the probe throw your breath, and then, or if you don't want to do it, give your blood or retina scan, and then it will tell you what are the chances, what disease you're having today, and what are the chances of you having other disease. Imagine. This is possible. It's also already happening in Delhi. There are tricorder-based test analysis being done at some places. Today, world-class analysis in health is available for free. 34 biomarkers for nearly any disease are available today. 54 biomarkers for any disease, and that's where I think things are moving so fast that it's unbelievable what will be the future of medicine tomorrow. Today, as it is, our lifespan has increased by 3 months per year. Every year, an average Indian will live 3 months more than what he lived last year. Today, it's very common to have people living 80 and 90. When India got independence, our lifespan was 40 or 42 years. And today, our average lifespan is 68 years. So imagine, life beyond 100 years, it's very much possible. Absolutely. Give another 10, 15 years, you will find many people living beyond 100 years. So this is all because of the advances that we have now in being able to diagnose a disease early before it sets in and to treat it before it causes a disease. Today, I may remain a strictly vegetarian, but still have a non-vegetarian meat. You know how? Because I don't need to create the meat from killing an animal. I can grow the meat from a single cell in the laboratory. It's actually sold today. Meat made in the laboratory. It's sold. It's available today. So like that, many such things are happening today. And that's where I think the entire health scenario, next slide please, in the days to come will change completely. Computers are getting much, much better in performance. This year, a computer beat the best Go player in the world. In 2030, computers will become more intelligent than humans. That is a prediction, although I don't know if that prediction will come true. But the fact is, computers and software and artificial intelligence are growing with such increased speed and enormity that today, you have heard of the taxi service Uber. Uber is the world's largest taxi network. तो आप सोचते होंगे कि इनके पास तो बहुत सारी गाड़ी होगी, इनके पास तो गाड़ियों की एक लंबी लाइन होगी. आपको ताज्जुब होगा, you will be very आश्चर्य होंगे आप कि उबर के पास एक भी गाड़ी नहीं है. Not even a single vehicle Uber owns. All it has is a tool, is a software, is an application app that you can download on your phone. And from that app, people who have their own car, they have linked with Uber. So the Uber will tell you, आप चले जाओ अभी यहाँ से स्टेशन पर, आपको एक सवारी वेट कर रही है. So you will also know where your taxi is, how much time it will take, to imagine how things have changed. Taxi stands have disappeared. Taxis have disappeared. They're all Uber. And yet they don't own a single taxi. दुनिया का सबसे बड़ा होटल चेन आप कहेंगे शायद ओबेरॉय है या ताज है, नहीं, या वेलकम ग्रुप है, नहीं, इट्स एयरबीएनबी. Airbnb is now the biggest hotel company in the world. इनके पास तो बहुत सारा होटल होगा पूरे दुनिया में. They don't own a single square inch of hotel space anywhere. All they have is an app that I can use. I've stayed in Airbnb hotels. All I do is I don't even know who the manager is. I don't even know who to give the key. An app comes to me and a message comes to me. The key is lying on that desk near your room. Go there and open. I open and see. And then once I'm done, I must clean up the place completely. If I don't clean up the place, next time when I try Airbnb, they will not give me because my record has gone. This person broke 5 glasses. Don't give him any hotel room. This person left the kitchen very dirty. Don't give him. So I have to be careful. And when I have to leave, all I have to do is my billing is done already on my iPhone. And the key, as I've given instruction, leave the key at that door. I don't know who came and picked up the key, who cleaned the house, nothing. We have to. So that is where technology is moving. So today, we have technology that can do everything. And tomorrow, I think softwares will be available thanks to machine learning and artificial intelligence, which will do everything that the human mind or human brain could never even think of doing it. Next slide, please. Next slide, please. So what are the challenges ahead? There are many, many challenges in engineering. Challenges such as polymer science, blood flow, bladder problem, fluid dynamics, and many other. When I joined IIT Delhi, I was wondering, what will I do? People used to ask me, you are a biologist, you are a medical microbiologist, what are you doing at IIT Delhi? I said, I'm trying to learn engineering from my engineering colleagues. And I spent almost a year trying to convince people that we must do biology-inspired engineering. Learn from nature. Learn from how nature does. Human body is an example of nature की जो उत्कृष्टियां हैं उसका उदाहरण है हमारा शरीर. क्या क्या चीज नेचर करता है जो हम उसका सोच भी नहीं सकते हैं. Why can't we learn from them nature and then do something engineering? And I'm very happy in a matter of 8 years that I stayed, we produced 2 technologies, and both these technologies have a huge promise. We have created a startup company, and this company is now working on these technologies, and now ICMR has adopted one of them. The other one is also going to be adopted, so that it can be used for a very quick and rapid diagnosis of TB when you don't have any electricity, any trained worker, any air conditioning, nothing of that sort, no infrastructure, yet you can diagnose TB better than any other existing technology today. We have already proven and showed that this actually works. The President of India was very kind to give us an award for this technology. And of course, we now are being funded by venture capitals to make this. So biology inspired in the next slide, please. Engineering is going to be. So where is the world heading? Next slide, please. The world today and tomorrow. Today, more than one biological mother, I told you, it's possible. You can be a child of 2 ladies. You can actually today, it's possible that you don't need to have sex to create a baby. You want to have sex only to enjoy, not for creating a baby. It's actually possible. So which means babies can be created by other mechanisms, not for sex. Disease-free babies, it's absolutely possible. Store your healthy organ for possible future use. Same pet as long as the owner lives. Dogs live for about 13, 14 years. Other pets live different times. But human life today, an average would be about 70, 80 years. You can have your same dog live with you for 80 years. It's possible. Contact lens with chips. No need to memorize. It's possible. This is all happening today, my friend. You don't have to wait for tomorrow. And tomorrow, it's going to be more and more difficult. There'll be a lot of things happening. But tomorrow's idea will all be based on a cell phone. No idea will work tomorrow. No technology will work tomorrow if it is not available on your cell phone. If any idea designed to succeed only if it is on cell phone. If it is not, it's doomed to fail. And that is where tomorrow is heading to. Next slide, please. There are still many issues, which is why I might chose the title that many more Nobel Prizes will come and all will be on biology. Let me give you a few examples. धोनी जी, कितना समय हमें आप और देंगी आप?
Time is all yours. How much you can spend?
ऐसा ना बोले आप कहेंगे तो मैं आप अगले 4-5 घंटे तक बोलता रह जाऊंगा.
Sir, we will be blessed to have you.
No, I'll take another 10 more minutes, not more than 10.
Yes, sir. Yes, sir. It's our opportunity to have you.
Let me give you the example of the human eye. You know, अगर हम आज के डेट में जो ह्यूमन आई जो काम कर सकती है, आप अगर मैग्नीफाइंग ग्लास लेते हैं या कैमरा लेते हैं तो कैमरा को शूट करने के पहले फोकस करते हैं ना. आई में कभी फोकसिंग करना पड़ता है? दूर की चीज देख रहे हैं वो भी साफ दिख रही है, नजदीक की देख रहे हैं वो भी साफ दिख रही है. It does auto focusing. Today there is no camera available that can do auto focusing. Likewise, many things. You can do the cropping, you can do a chopping, you can do, the amount of the power that's sent to the camera sensor at any given time is unbelievable by the human eye. And whereas for the artificial camera or artificial thing, there's no way you can do it. Yet there is no camera that can do multi-focusing without blurring of background images. And if I have to try to create a camera similar to what a human eye does, it will cost me about $35 million for one camera to be created. सोचिए भगवान ने कितने अच्छे नेत्र हमें दिए, हम उससे सब काम कर लेते हैं और एक नया पैसा मुझे नहीं चार्ज हो रहा. और वो सब काम जो हम कर रहे हैं कोई कैमरा आने वाला नहीं कर पाएगा. दूसरा उदाहरण मैं देता हूं. नेक्स्ट स्लाइड. दूसरा उदाहरण जो है, नेक्स्ट स्लाइड प्लीज. हम अपने आपने देखा होगा कि कभी कभी आपका हाथ में अगर कुछ कट जाता है तो थोड़ी देर के लिए उसमें से लहू निकलता है, लेकिन उसके बाद सूख जाता है और उसके बाद वो ब्लड निकल जाता है, ऊपर से नया चमड़ा ग्रो कर जाता है. ये क्यों होता है? बिकॉज वी हैव अडल्ट स्टेम सेल्स एंड दी स्टेम सेल्स अगेन क्रिएट. लेकिन कभी ये नहीं होता कि अगर आपकी उंगली कट गई किसी कारण से तो नई उंगली जुड़ गई. लेकिन अगर आप एक छोटा सा मकड़े को लेंगे, उसका टांग अगर टूट गया तो थोड़ी देर के बाद वो नया टांग वहां बना लेता है. उसको कहते हैं रीजेनरेशन. मकड़े कर सकते हैं, हम लोग क्यों नहीं कर सकते? अगर हम रीजनरेशन कर सकते तो मैं अपने शरीर में ऐसा रीजनरेशन कर लूं कि मैं कभी बूढ़ा लगूंगा ही नहीं. तो दैट इज something which we have no clue on it. People are trying, and that can also reverse the aging process. Tadpole की स्टोरी आपने सुना होगा, उसकी एक पूंछ होती है. After some time, the पूंछ drops out. It loses it. जब हम पैदा होते हैं तो ये हमारी पांचों उंगलियां एक साथ होती हैं. आहिस्ते आहिस्ते इनके बीच के सेल्स मर जाते हैं और मर करके ये पांचों उंगलियां अलग हो जाती हैं. तो can we allow this to be reversed? And that is where people are now looking at how can we do regeneration. Next slide, please. This is a big question today among biologists. Brain की बात करें, मस्तिष्क की. और brain consists of about 100 billion neurons. 1 बिलियन में 9 शून्य होता है, मिलियन में 6 शून्य होता है. 1 पर 10 के पर 6 शून्य लगा दीजिए तो 1 मिलियन हो जाता है. 1000 में 3 शून्य होता है, 1 पर 3 जीरो लगाएंगे तो 1000 हो जाएगा. बिलियन में 9 शून्य होता है. तो हमारे ब्रेन में 100 बिलियन न्यूरॉन है. दैट इज अबाउट 165 टाइम्स द नंबर ऑफ पीपल ऑन दिस प्लैनेट. ऑल सच लिविंग इन आवर ह्यूमन ब्रेन. थॉट प्रोसेस, अभी मैं आपसे बात कर रहा हूँ, मेरा ब्रेन भी काम कर रहा है. जो मैंने सुबह में भोजन किया है उसको वो डाइजेस्ट कर रहा है. हमारे बोलने के लिए लिप मूवमेंट करा रहा है, हमारी आंख का मूवमेंट करा रहा है, हमारी हैंड मूवमेंट करा रहा है, हमारी सोचने की प्रक्रिया को चालू रख रहा है, हमें सांस भी लेने. इमेजिन कितनी सारी चीज मल्टीटास्किंग कर रहा है. ऑल हैज टू बी डन साइमल्टेनियसली. ये नहीं कि रुक जाओ, पहले हम बोलना लिप बाद में कर देते हैं, बोलेंगे बाद में. ये नहीं, इट हैज टू बी डन साइमल्टेनियसली. इमेजिन हाउ मच मल्टीटास्किंग कैन इट बी डन. तो आप कहेंगे कि इतना ज्यादा मल्टीटास्किंग जब करना होगा ब्रेन को, ये तो बहुत ज्यादा एनर्जी लेता होगा. आज के हम लोगों जब when we were advising the prime minister, we suggested that we should have a super computer, because उन दिनों में भारत को सुपर कंप्यूटर के लिए परमिशन नहीं दिया था, ब्लैकलिस्ट कर दिया था, दीवार कर दिया था. We said we'll create one. समस्या वहां खड़ी हुई जब पता लगा उस सुपर कंप्यूटर को चलाने के लिए जो बिजली की जो क्षमता चाहिए, बिजली की जो आवश्यकता चाहिए, वो तो अनबिलीवेबल है. फिर भी वो सुपर कंप्यूटर वो काम नहीं कर पाएगा जो हमारा मस्तिष्क करता है. मस्तिष्क कितना पावर कंज्यूम करता है? मात्र 10 वॉट. सिर्फ 10 वॉट पावर में हमारा ह्यूमन ब्रेन वो कुछ कर देता है जो आज का सुपर कंप्यूटर कभी नहीं कर पाएगा, कर पाता है, न कर पाएगा. So that is where we need to figure out. Can we not learn how the brain excels? Next slide, please. Last example I'll give you. हमारा हृदय दिन भर में तकरीबन 80,000 से 1,00,000 मरतबे धक धक करता है. एक साल में 35 मिलियन बार ये धक धक करता है और कभी रुकता नहीं है. एवरेज लाइफ टाइम में 2.5 बिलियन टाइम्स द हार्ट बीट्स. आप कोई ऐसा औजार, कोई ऐसा यंत्र दिखा दें जो बगैर रुके लगातार काम करता जाए. हमारे शरीर में तकरीबन 5-5.5 लीटर ब्लड है, 5.6 लीटर लहू है, और ये ब्लड पूरे शरीर में 3 बार 1 मिनट में सर्कुलेट होता रहता है. जरूरी है क्योंकि ब्लड के साथ ऑक्सीजन होता है. ऑक्सीजन आप शरीर की आवश्यकता है. अगर सर्कुलेट नहीं करेगा तो ऑक्सीजन शरीर के डिफरेंट सेल्स को नहीं मिलेगा. तो ये आवश्यक है और ये हमेशा करता रहता है, कभी रुकता नहीं है. थोड़ा स्लो कभी हो जाता है, लेकिन कभी हम ज्यादा एक्टिव हो जाते हैं तो ज्यादा तेजी से करता है. कौन बताता है इसको? कैसे हार्ट समझ लेता है? मात्र एक दिन में ब्लड 19,000 किलोमीटर की दूरी तय करता है. अगर आप पूरी दूरी नापें यहाँ से कहाँ जाता है तो 19,000 किलोमीटर एक दिन में ब्लड, यानी कि 12,000 माइल एक दिन में ब्लड तय करता है. जो पूरे विश्व की भ्रमण करेंगे आप, एक जगह से निकल के वापस वहीं आएंगे, वो भी इतना नहीं है. इमेजिन कैसे कर लेता है. कैन वी नॉट लर्न फ्रॉम द ह्यूमन हार्ट? आज के डेट में कोई आप पंप बना सकते हैं जो इतना काम करे और उसको रोके कभी नहीं? 100 साल तक वो पंप काम करता रहेगा बिना रुके एक दिन. ऑफ कोर्स कोई डिजीज होगी तो बात अलग है. इमेजिन, दिस इज वेयर वी मस्ट लर्न फ्रॉम नेचर. And that's what I've been trying to convince, and I'm very happy that many of my scientists today, IIT Delhi has one of the best biology program today are in IIT Delhi. And I think where people are learning that we must learn from nature. Next slide, please. Today, we are now moving into an era where we have this virus which causes the disease known as COVID. Next slide, please. And COVID has not left any country of the world. हमारी सभ्यता में हम लोग को सिखाया गया था, मैंने संस्कृत पढ़ा था 10वीं कक्षा तक. हमारे पिताजी ने कहा कि आप भारतीय हो, आपको एक संस्कृत का पंडित रख करके उन्होंने हमें संस्कृत का ज्ञान दिया. और हमें बड़ी खुशी है कि मैं संस्कृत के श्लोक भी जानता हूँ और संस्कृत पढ़ता भी हूँ. थोड़ा बहुत समझने की कोशिश करता हूँ, लेकिन अब तो नहीं समझ सकता हूँ, पाता हूँ क्योंकि व्यवहार कम हो गया. लेकिन संस्कृत में एक हमें पढ़ाया गया था वसुदेव कुटुंबकम, द वर्ल्ड इज अ फैमिली. अब ये कोविड ने साबित कर दिया कि वसुदेव कुटुंबकम, पूरे विश्व का कोई भी प्रांत, कोई भी चप्पा नहीं है जहां कोविड ने अपनी जो बर्बादी नहीं दिखाई है. कोई भी प्रांत, कोई भी स्क्वायर इंच, हर जगह कोविड की प्रॉब्लम शुरू हुई. जहां जहां ह्यूमन रेस रह रहे हैं, जहां जहां मानवता रहती है, क्योंकि मानवता ही को पता लगता है कि कोविड ने क्या कर रहा है. तो कोविड ने दिखा दिया कि वर्ल्ड इज अ फैमिली. इकोनॉमी का सत्यानाश किया कोविड ने, सभी दुनिया की इकोनॉमी, सिर्फ हम ही लोग के नहीं, सभी दुनिया की. तो ये कोविड की, इट हैज बीन रिस्पॉन्सिबल फॉर सेटिंग, नेक्स्ट स्लाइड प्लीज, अ न्यू लाइफस्टाइल फॉर ऑल ऑफ अस. अब कोविड में जैसे जैसे ये वायरस, अब मैंने शुरू में बताया था कि कोविड इज द स्मार्टेस्ट पैथोजन नोन टू ह्यूमैनिटी सो फार. कल को हो सकता है कोविड से भी भयंकर पैथोजन आ जाए, वी डोंट नो, संभव है. लेकिन आज के डेट में कोविड जैसा भयंकर पैथोजन किसी ने नहीं देखा. स्मार्टेस्ट पैथोजन है और ये इतना तेजी से इवॉल्व कर रहा है. इतना तेज इवॉल्व करने वाला, इवोल्यूशन इज अ स्लो प्रोसेस ड्रिवन बाय नेचर. यहाँ नेचर तो हमारा शरीर है, हमारा शरीर इसको इतना तेजी से इवॉल्व करा रहा है कि बहुत तेजी से अलग अलग जगहों पर, अलग अलग विश्व के कोनों में ये वायरस अलग अलग तरह से इवॉल्व कर रहा है। इंडिया में जो इवॉल्व किया था लास्ट वायरस, उसको डेल्टा वायरस का नाम दिया गया। अभी डेल्टा वायरस इतना ज्यादा घातक वायरस है, इतना ज्यादा कि बहुत तेजी से फैलता है। तो इस वायरस को पता है कि कहाँ पर मुझे अपने अपने शरीर में, अपने अपने आरएनए में परिवर्तन लाना है, ताकि मैं और भी बलवान हो जाऊं, ताकि मैं और भी तेजी से लोगों को इन्फेक्ट करूं, ताकि और भी मैं वैक्सीन का असर ना हो हम पर। So that is where we have to learn from these viruses, how they are evolving, and what is the basis for the next slide, please, for them to evolve so rapidly and so effectively. So this evolution in turn increases the transmissibility and increases the virulence or the change in the clinical disease presentation. पहले होता था कि इससे सिर्फ लंग में इन्फेक्शन होता था, आज पता है कि लंग ही नहीं, हार्ट फेलियर भी बहुत लोगों को हो रहा है। तो ये कैसे कैसे परिवर्तन ला रहा है, ये कोविड का वायरस करा रहा है, कैसे करा रहा है। एंड दिस इज ऑफ कोर्स मैनिफेस्टिंग इन मेनी थिंग्स। एंड टुमारो माय फियर इज दैट हमने वैक्सीन तो ले लिया है, आप लोगों ने अगर नहीं लिया जिनमें से, तो मैं तो बहुत आपसे हाथ जोड़ के विनती करूंगा कि आप वैक्सीन ले लें। वैक्सीन लेना अनिवार्य समझिए। वैक्सीन से ही कुछ हमारा बचाव अगर हो सकता है तो वैक्सीन से ही होगा। आज के डेट में कोई दवा नहीं है कोविड के लिए। जो दवा है वो सिर्फ एक्सपेरिमेंटल मेडिसिन है जो ट्राई की जा रही है कि चलो देखते हैं काम करता है तो वाह वाह, लेकिन वैक्सीन से उम्मीद है कि हमें प्रोटेक्शन मिलेगा। तो बट उसकी भी उम्मीद की भी जिस घातक रूप से ये वायरस म्यूटेट कर रहा है, हम 100% नहीं कह सकते हैं, बट स्टिल कुछ तो प्रोटेक्शन है, उम्मीद तो है। नेक्स्ट स्लाइड प्लीज। सो इवॉल्विंग स्मार्ट वायरस, एक नया टर्म WHO ने निकाला था, वेरिएंट्स ऑफ इंटरेस्ट। इंटरेस्ट मतलब भाई ये तो वेरिएंट हो रहा है, शायद ये वेरिएंट को कुछ संभावना है कि कुछ गड़बड़ कर सकता है। तो जब गड़बड़ करने की संभावना स्थापित हो जाती है तो इट बिकम्स वेरिएंट्स ऑफ कंसर्न। ये हम लोगों ने अभी लास्ट मंथ एक पेपर पब्लिश किया नेचर मेडिसिन में, where we have characterized the variants of concern and including the Delta variants that came out of India. And we have shown what all these variants can do. Of course, this paper is being followed up by another paper that we again communicated to another big journal. We don't know. So this is where the virus is. Next slide, please. I'm not getting into more details. जिनको इच्छा होगी वो मुझे लिख सकते हैं या पेपर को पढ़ सकते हैं, तो they can find out more. So actually कभी-कभी हमें सोचना लग पड़ता है कि ये वायरस कैसे काम करता है। ये देखता है कि कहाँ-कहाँ शरीर में ब्लाइंड स्पॉट है। It looks for what are the vulnerable, who are the vulnerable people, where are the blind spots, and so that it can cause high degree of transmission. And despite our efforts, पूरी कोशिश करते हैं कि हमें ये वायरस का इन्फेक्शन ना हो, फिर भी बेनिफिट कर देता है। लेकिन एक चीज मैं अवश्य कहूंगा आप सब लोगों को जो भी हमारा लेक्चर सुन रहे हैं, कि मास्क का उपयोग अवश्य करें। अभी तो इस रूम में हमारे पास कोई भी नहीं है, इसलिए मैंने मास्क निकाल दिया है। अदरवाइज मास्क के उपयोग से ये सिद्ध हो चुका है कि वायरस का आप इंजेक्शन जो आप करेंगे, इनहेलेशन करेंगे, उसमें काफी कुछ रुकाव हो सकता है, बचाव हो सकता है। तो सारांश में मैं ये कहूंगा कि our knowledge about the virus is changing all the time. और कभी कभी हमें बड़ा विवशता लगती है कि हम क्या हैं। What is it? Is SARS virus actually that smart? Or is it just that humans are tragically stupid? हम लोग अपने को सबसे उत्कृष्ट जो प्रकृति ने बनाई उनमें से हम अपने को समझते हैं, most exalted creation of man, of nature. Look at the small virus, we can't even see. Just 30,000 nucleotides. वो भी single stranded RNA. And it is making us literally cry every day. So many of our lives, our relatives are lost, and we don't know, maybe the third wave will come. We don't know what will happen. So this is where this virus, next slide please, is causing a repeated attack. So we are really racing against time. The third wave of COVID-19 variants we are spreading. COVID has already changed the world we live today. अब पढ़ाने का जो प्रक्रिया होती थी, पहले आई टू आई कांटेक्ट होता था, बच्चे हमारे सामने बैठते थे, मैं भी लेक्चर देता हूँ, मैं भी पढ़ाता हूँ, वो आनंद अब नहीं मिल रहा है। अब इस पर आप लिख करके अब आपने अपना कैमरा ऑफ कर दिया, मुझे पता ही नहीं कि आप सुन भी रहे हैं या कहीं और चले गए। आज के डेट में ये संभव हो गया हमारे जैसे लोगों के लिए कि दिन भर में 3 मीटिंग 4 मीटिंग इंक्लूडिंग इंटरनेशनल मीटिंग अटेंड करते हैं, लेक्चर देते हैं, क्योंकि वहां जाना नहीं होता। एक मीटिंग का समय 2 घंटे का मॉर्निंग का समय है, 2 घंटे के बाद कहीं और समय है। अभी हमारी ब्रिक्स की मीटिंग थी, आई एम चेयरिंग दैट मीटिंग, तो रात में उसकी मीटिंग शाम में 9 बजे से शुरू हुई, क्योंकि ब्रिक्स में ब्राजील, रशिया, इंडिया, चाइना, साउथ अफ्रीका, तो उनका सबका टाइम जोन अलग अलग है, विभिन्न है, तो उनको अकोमोडेट करने के लिए रात में 9 बजे शुरू हुई और 11:30 तक खत्म हो गई। तो सुबह 9 बजे से 11:30 बजे के बीच में हमने 5 मीटिंग 4-5 मीटिंग तो कर लेते हैं। ये तो कोविड की देन है, लेकिन इसकी जो भयानक रूप जो हम लोग को देखने को मिल रहा है, उससे पता नहीं हम लोग कब तक बच पाएंगे या बच भी पाएंगे कि नहीं पाएंगे। एंड कैन वी बी श्योर कि कोविड-19 के बाद अब कुछ नहीं आएगा? नहीं कह सकते, वी कैन नॉट बी श्योर। लेकिन इतनी बात तो सही है कि आने वाले एटलीस्ट नेक्स्ट 4-5 साल कोविड पर इतना शोध होगा, इतना ज्ञान उस पर मिलेगा, लेकिन हर बार लगेगा कि ये तो नई चीज पता चल रही है। आप पढ़ते होंगे कि कभी पहले हुआ कि कोविड ऐसे स्प्रेड करता है, अब पता लगा नहीं कोविड ऐसे स्प्रेड करता है। पहले पता लगा कोविड ओनली लंग्स में जाता है, सबको वेंटिलेटर पर डालना पड़ता है। अब पता लगा लंग्स पर नहीं जाना, लंग्स पर जाता है बट वेंटिलेटर की जरूरत नहीं है। You know, everything is transforming and we don't know what all it can do. Next slide, please. And that is where I think we are changing. अगर आपके पास समय, अगर हमें 5 मिनट मात्र और दे दें, मैंने 1 घंटा ले लिया पूरा, चूंकि नहीं 1 घंटा नहीं 45 मिनट लिया है, चूंकि 11:15 पर शुरू किया था, नहीं 1 घंटा ले लिया। आई एम सो सॉरी, मैंने 1 घंटा ले लिया।
Sir, that's absolutely fine. We are actually very opportunity. We are finding this opportunity to listen you. Nothing can be bigger than that.
चलिए तो मैं आपका 5 मिनट मात्र और लूंगा। ये बड़ा अच्छा प्रश्न मैं
Sir,
अपने आप
all
से कर रहा हूँ, अपने आप से कर रहा हूँ। और ये प्रश्न ये है कि कैन वी लुक एट अ मोर फंडामेंटल इशू। हम लोगों ने सबने पढ़ा था कि डीएनए बड़ा प्राथमिकता उसका स्थान है। डीएनए आरएनए बनाता है, आरएनए प्रोटीन बनाता है, और प्रोटीन से फिर फंक्शन होता है, फंक्शन से फिर जीवन बनता है। अलग अलग प्रोटीन चाहिए अलग अलग जीवन के काम करने के लिए। इसमें भी क्वेश्चन मार्क जो लगा हुआ है, आरएनए भी फंक्शन देता है, प्रोटीन भी फंक्शन देता है। पहले हम लोग समझते थे कि एक प्रोटीन से एक ही फंक्शन, नोबेल प्राइज मिल गई इस काम पर, 1 जीन 1 प्रोटीन हाइपोथेसिस। आज के डेट में अब हमसे पूछें आप, तो मैं कहूंगा ये गलत है। 1 जीन डज नॉट गिव यू 1 प्रोटीन। 1 जीन कैन गिव यू गला इतना ज्यादा, मैंने 1.25 घंटा लेक्चर लगातार कभी नहीं दिया था, तो इसलिए शायद गला कसरत कर रहा है, बट एनीवे आई विल बी फाइन। तो बट वी नाउ नो देयर आर नो रीजन्स इन द प्रोटीन नोन एज इंट्रिंसिकली डिसऑर्डर्ड रीजन। मतलब एक ऑर्डर्ड होगा तो एक की एंड लॉक का स्ट्रक्चर आपने पढ़ा होगा। प्रोटीन का एक लॉक होता है, उसकी एक की होती है, और लॉक एंड की स्ट्रक्चर से ही फंक्शन होता है। नाउ वी नो नो लॉक एंड की स्ट्रक्चर। They can adopt any structure depending, because it's no defined structure, disordered. और फिर वो डिसऑर्डर से लाइफ कैसे बनी, ये सबसे बड़ी समस्या। नेक्स्ट स्लाइड प्लीज। और ये समस्या शायद वैज्ञानिकों को अगले 400-500 साल तक, अगर ये हमारी दुनिया टिकी रही 400-500 साल तक, ये सवाल करती रहेगी, जीवन क्या है, किसको हम जीवित कहते हैं, जीवित की परिभाषा क्या है। जीवित की 3 परिभाषा है। एबिलिटी टू रेप्लिकेट, अपने जैसा दूसरा पैदा कर देना, सिमिलर काइंड, रिप्रोडक्शन। दूसरी परिभाषा है ग्रोथ, उसको थर्मोडायनेमिकली कहते हैं रिडक्शन ऑफ इंटरनल एंट्रोपी एट द एक्सपेंस ऑफ एनवायरमेंटल एंट्रोपी। और तीसरी जो बिल्कुल आवश्यक परिभाषा है, मेटाबॉलिज्म, मेंटेन लोअर इंटरनल एंट्रोपी। किसी भी चीज में अगर ये तीनों चीजें नहीं हैं तो उसे आप जीवित नहीं कह सकते। उसको जीवित की भाषा में उसको नहीं गिन सकते हैं। और जीवित होने के लिए उसका जो अगर आप केमिकल डिसेक्शन करें तो 6 एलिमेंट हैं, तत्व हैं, कार्बन, हाइड्रोजन, ऑक्सीजन, नाइट्रोजन, फास्फोरस, शुगर। किसी भी जीवित प्राणी में, चाहे वो बैक्टीरिया हो, वायरस हो, उसमें आप ये 6 तत्व अवश्य पाएंगे। इसके अलावा इन्हीं का अलग अलग मिश्रण होगा, लेकिन ये 6 प्राथमिक जो तत्व हैं ये अवश्य मिलेंगे आपको। नेक्स्ट स्लाइड प्लीज। तो सब सवाल पूछा जाता है, नेक्स्ट स्लाइड प्लीज। हाउ डिड लाइफ बिगिन? आप अपना अलग आपका धर्म है, मेरा अलग धर्म है। हम लोग मानते हैं कि हम ईश्वर ने अपनी अपनी रचना की जीवन की। कुछ लोग कहते हैं कि नहीं, हम ईश्वर ने नहीं किया, हम तो इवॉल्व किए हैं। मैं उस पर नहीं करूँगा। मैं एक फंडामेंटल सवाल पूछता हूँ कि आखिर जीवन की शुरुआत कैसे हुई? फ्रांसिस क्रिक, जिनको कहा जाता है ही इज द आइंस्टाइन ऑफ बायोलॉजी, जैसे आइंस्टाइन का जो दर्जा था, उनका जो स्थान था साइंस में, वही स्थान फ्रांसिस क्रिक का था। ही इज नो मोर। उन्होंने अपनी किताब में लिखी है, एन ऑनेस्ट मैन armed with all the knowledge available to us now could only state that in some sense the origin of life appears at the moment to be almost a miracle. So many are the conditions which would have had to have been satisfied to get it going. मतलब फ्रांसिस क्रिक जैसे आदमी भी, जो कि आइंस्टाइन के तरह जाना जाता है उनको, वो भी मानते हैं कि ये कैसे जीवन की प्रक्रिया कैसे शुरू हुई, ये तो एक चमत्कार है। एंड चमत्कार कैसे हुआ? तो लिविंग ऑर्गेनिज्म, मेरा मानना है, एक नेचुरल बायो कंप्यूटर है, जिसमें हार्डवेयर अलग है एंड सॉफ्टवेयर अलग है। ये जो मैं बता रहा हूँ, ये मैं अपनी 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 सोच बता रहा हूँ, ये आपको किताब में कहीं नहीं मिलेगी। जो कि आपके सुनने वाले छोटे छोटे बच्चे भी होंगे, विद्यार्थी भी होंगे और कलीग्स भी होंगे। I hope to activate and hope to impress some people that there is a reason to think beyond what you find in the books and do something very different. और ये मैं बचपन से सोचता रहता था कि कैसे जीवन की परिभाषा कैसे कैन बी, जीवन शुरुआत कैसे हुई। तो नेक्स्ट स्लाइड प्लीज। तो मैं मानता हूँ कि हमारा जीवन जो है वो एक लाइफ इज नथिंग बट अ नेचुरल कंप्यूटर। अभी तक तो सब लोग, हम लोग सब समझते हैं कि डीएनए ही सब कुछ है। ऑन अ जेनेटिक लेवल ह्यूमंस एंड बंदर और अब सेम डीएनए है, मोर देन 96% ऑफ द डीएनए इज कॉमन। तो फिर वो और हम इतने विपरीत क्यों हैं, इतने अलग क्यों हैं? केमिकल स्ट्रक्चर कैरीज अ पर्टिकुलर केमिकल इंफॉर्मेशन। वो सब में से यही 6 मॉलिक्यूल 6 एलिमेंट्स हैं, यही से स्ट्रक्चर बनाते हैं। मेरा मानना है कि डीएनए जो नेचुरल बायो कंप्यूटर है, उसका हार्डवेयर इज डीएनए। जब मैं इंडिया वापस आया अमेरिका से, टू कम बैक एंड लिव इन इंडिया, मुझे याद है मैं एक बार अपने घर आया था, इंडिया आया था व्हेन आई वाज वर्किंग अब्रॉड। तो जब मैं वापस जाना चाह रहा था अमेरिका, आई वाज अबाउट टू गो डे आफ्टर टुमारो, देन माय फादर आस्क्ड मी नाश्ते के टेबल पर कि बेटा तुम्हारा क्या प्रोग्राम है? मैंने कहा पिताजी हम अभी अमेरिका वापस जा रहे हैं, अभी रहेंगे वहाँ 7, 8, 10 साल, उसके बाद सोचते हैं क्या करना है। तो बोले कि बेटा बहुत अच्छा है, जो भी करो अच्छा करोगे, आई एम श्योर, आई नो यू मस्ट बी डूइंग वेरी वेल, अच्छा कमा रहे हो, अच्छा खा रहे हो, सब कुछ है, स्टैंडर्ड वगैरह सब कुछ अच्छा है। एंड वी कम फ्रॉम अ वेरी एवरेज फैमिली। माय फादर वाज अ टीचर। तो उन्होंने कहा बेटा एक बात याद रखो, जंगल में मोर नाचा किसने देखा? ये तुम्हारा देश है, हम भारतीय हैं। यू मस्ट कम बैक टू योर कंट्री एंड गिव टू योर कंट्री। यहाँ लोगों को आप प्रोत्साहित करोगे, आप इंस्पिरेशन बनोगे लोगों के लिए। यहाँ जाओगे तो लोगों का, हमारे भारतीयों का कल्याण होगा। वहाँ जाके आप क्या कर लोगे? ठीक है, पैसे बहुत कमा लोगे, कुछ 10-15 लोग, कुछ अवार्ड भी मिल जाएगा, लेकिन यहाँ जो किसी को क्या, कैसे आपसे इंस्पिरेशन मिलेगा? कम बैक टू योर कंट्री। और ये शब्द, ये उनका जो जंगल में मोर नाचा किसने देखा, मेरे मन में बस गया। और विद इन 2 years आई डिसाइडेड, मेरे सुपरवाइजर ने कहा कि आप ग्रीन कार्ड ले लो। मैंने कहा ग्रीन कार्ड नहीं लूँगा। ग्रीन कार्ड लेने का मतलब है जब मैं इंडिया जाऊँगा, वहाँ जब जंग करूँगा, लड़ाई करूँगा सिस्टम से, पता लगा दूध नहीं मिल रहा है, बच्चे का एडमिशन नहीं हो रहा है, बिजली नहीं है, पानी नहीं है, मैं कहूँगा चलो लेट्स गो बैक टू अमेरिका यार, फॉरगेट इट। और हमारे जैसे लोग जिन लोगों ने ग्रीन कार्ड लिया था, ज्वाइन किया था मेरे साथ, नन ऑफ देम आर इन इंडिया, दे ऑल गॉन बैक। मैंने कहा ग्रीन कार्ड नहीं लूँगा। and I'm very happy that I came back. I listened to my father. तो अभी जो हमने जो कुछ कर पाए हैं, मैं सोचता हूँ कि हमारे साथ जो लोग वापस अमेरिका में रह गए, भगवान ने बड़ा दिया, हमें बहुत कुछ दिया है। सबसे बड़ी बात है कि हमें अपने भगवान, अपने जो माँ बाप हैं, उनको सेवा करने का मौका दिया। दे बोथ डाइड, दे बोथ लिव्ड विद मी। हम लोग 3 भाई बहन, 4 भाई बहन थे, सबको छोड़ के वो हमारे साथ रहते थे। यह बहुत बड़ी उपलब्धि है, बहुत बड़ी उपलब्धि है। और शायद जो कुछ मैं कर पाया हूँ उनकी वजह से, उनका जो मुझे आशीर्वाद मिलता रहा। तो इसलिए मैं आप सब लोगों को कहूँगा कि आप अपने माँ बाप को कभी ना छोड़ें। आप अपने माँ बाप की सेवा अवश्य करें, और जितना हो सकता है, जितना आप कर सकते हैं, उससे ज्यादा बढ़ चढ़ के करें। और कभी ये मत जताएं कि आप उनके लिए कुछ कर रहे हैं। आप कुछ नहीं कर पा रहे हैं, उन्होंने आपको जन्म दिया, आपको इस लायक बनाया। तो ये कमिंग बैक टू जीवन क्या है, व्हाट इज लाइफ, मेरा मानना है कि, और डीएनए इज अ हार्डवेयर। सॉफ्टवेयर क्या है? कंप्यूटर हम जब लेकर आए थे तो एक सिर्फ सॉफ्टवेयर डीएनए हार्डवेयर लेकर आए थे। यहाँ उन लोगों ने उसमें एक ऑपरेटिंग सिस्टम लगा दिया। अभी भी आप जो कंप्यूटर यूज करते हैं, तो हम तो मैक है, तो मैक ऑपरेटिंग सिस्टम है। आप कुछ और ऑपरेटिंग सिस्टम यूज करते हैं, मैक नहीं है। अगर ऑपरेटिंग सिस्टम हटा दें तो कंप्यूटर काम कर सकता है? यूजलेस, कोई फायदा नहीं है। नेक्स्ट स्लाइड प्लीज। तो डीएनए सब कुछ नहीं है, डीएनए इंपॉर्टेंट है। नेक्स्ट स्लाइड। तो जीवन और मृत्यु क्या संबंध है दोनों में? हमारे माता पिता तो भगवान को प्यारे हो गए। बड़ी खुशी की बात है कि हमारे पत्नी के माता पिता भी हमारे साथ ही रहते थे। वो भी हमारे नजरों के सामने वो गए हमारे साथ, हम दुनिया से विदाई लिया। यदि वाइफ की मदर एंड फादर, मदर वॉज एडमिटेड टू द हॉस्पिटल एंड आई कुड सी हर ECG going up and down and suddenly becoming flat. मैंने कहा क्या हो गया? बॉडी वही है, अब वो नाम उनका नहीं होगा, उसका नाम उनका बॉडी हो गया। ये क्या परिवर्तन आ गया उनमें? हम उनको कहते हैं उनकी आत्मा निकल गई, उनका सोल निकल गया, उनका रूह निकल गया। सेम डीएनए, सेम पर्सन, क्या हो गया उनको? व्हाट इज सोल? सोल को मैंने नाम दिया है लाइफ ऑपरेटिंग सिस्टम। ये लाइफ ऑपरेटिंग सिस्टम है मेरा, ये सिर्फ भगवान को पता है, ईश्वर को पता है। वही जानता है कि लाइफ ऑपरेटिंग सिस्टम क्या है। हम बिल्कुल स्वस्थ हैं, हमारा अपना नाम है। कल मैं मर जाऊंगा तो मुझे नाम से नहीं कहा जाएगा, वो कहा जाएगा बॉडी को उठाओ यहां से। कोई नहीं कहेगा कि हसनैन को उठाओ यहां से, बॉडी को वहां रखो, बॉडी को वहां ले जाओ। क्या हो गया? सेम, सब, एवरीथिंग इज आइडेंटिकल, बिकॉज आपका जो ऑपरेटिंग सिस्टम है भगवान ने निकाल लिया आपके शरीर से। ये लाइफ ऑपरेटिंग सिस्टम क्या है? इफ वी नो दैट, that is $1 zillion question. And that is where my belief is that biologists and physicists and chemists and mathematicians and information technologists will all be looking at trying to identify this life operating system, which is known only to the Creator. Thank you so much. Last slide. Last slide. I have a big lab, as धोनी जी ने बताया. I have a big lab, big group. मैंने अपने साइंस के बारे में कुछ नहीं बताया, थोड़ा बहुत खाली काम हमने कोरोना वायरस पर जो किया है वो बताया. And I am very happy कि Government of India has been supporting me very generously and continues to support me. 2026 तक हमारे पास काफ़ी अच्छे ग्रांट हैं. I have a good number of people who I collaborate with, including from India and abroad, including हमारी वाइफ जो डॉक्टर नसरीन ज़फ़र एहतशाम है, वो डायरेक्टर थी नेशनल इंस्टीट्यूट ऑफ पैथोलॉजी. अब वो भी रिटायर कर गई हैं एंड शी इज़ नाउ बिकम एमेरिटस साइंटिस्ट. उनके साथ काफ़ी हमारा कोलैबोरेशन है, कंबाइंड लैब है. एंड डॉक्टर मदन बाबू, मेनी अदर्स हु हैव बीन वर्किंग टुगेदर. और तकरीबन 150 से ऊपर लोग, बच्चे हमारे साथ काम कर चुके हैं. उसमें कई को MD मिली है, कई को PhD मिली है, कई ने पोस्ट डॉक किया है. और ये जो लेफ्ट साइड पर आप नीचे देख रहे हैं IMC स्क्वायर एंड CTB, ये दोनों, these two are new technological platforms that we work together with IIT Delhi, and this is something which we believe will create a major paradigm shift in the way TB and other diseases are diagnosed. That's where we are. Last slide. थैंक यू सो मच फॉर बीइंग हियर। अगर हमारे लिए कोई भी प्रश्न हो तो हमारे वेबसाइट पर चले जाएं sayyidehasnain.org.in और मैं कोशिश करता हूं कि हर एक प्रश्न का मैं उत्तर देता हूं। और ध्वनि जी से एक अनुरोध है कि वो हमारा जो ईमेल आईडी है sehiittd@gmail.in, मैं फिर से बताता हूं SEH मेरे नाम का इनिशियल वर्ड है, सैयद एहतेशाम हसनैन, IITD इंडियन इंस्टीट्यूट ऑफ टेक्नोलॉजी दिल्ली, उसका संक्षेप में है @gmail.com। इस पर मुझे कोई भी आप मेल भेजेंगे, मैं कोशिश पूरी करता हूं कि हर एक मेल के जवाब मैं खुद से दूं, और मैं दूंगा जवाब। कोई भी समय बहुत ले लिया मैंने आप लोगों का।
No, sir, not at all. We are very much fortunate to have you.
सवा घंटा टाइम ले लिया मैंने, but thank you so much.
Thank you so much, sir. We are really very fortunate to hear you in person. And I'm very much sure that all the viewers are astonished, encouraged, and gained a lot from your instructional and exploratory sessions, sir. I finally request our Honorable Provost, sir, to provide some thank words to sir. Please, sir.
मुझे बड़ी बड़ी प्रसन्नता है कि हमारे वाइस चांसलर साहब को उतना समय है कि वो बैठे रहे, सुनते रहे। This is an indication of the respect that he has for science. I really admire you and I want to salute you, Jai Hind, for doing such a wonderful thing. You know, it's very rarely you find somebody at your level to spare time. Normally, हम जैसे लोग इनॉग्रेट करके चले जाते हैं, सुनने को वेट नहीं करते हैं, लेकिन आप मुस्तकिल बैठ के सुन रहे थे। बहुत बहुत धन्यवाद, बहुत बहुत
And he is very cooperative. Once when I invited and that sir is coming, he himself asked me thrice that when are you planning? I just want to attend this, and very
Thank you so much.
warmly he is accepted.
Sir, it's really a pleasure, and I was hearing you because something was very interesting for me to learn even. No doubt I have passed through 2-3 universities, Vice Chancellor, the big two state university of Gujarat, that Gujarat University and Gujarat Technological University. I used to be the Vice Chancellor there. I was instrumental to make digitalization of the entire Gujarat as far as admission and other activities are concerned. After the retirement from the government, I am here since last 4 years, and here also we are doing something new. And people like you, when we are getting as a resource person, I also take advantage to learn from that. Otherwise, there are so many activities going on, and I have 2-3 devices on my table. So I'm changing that. So in some where I have to speak, I just to connect that, and then I'm hearing people like you. And that's why every half an hour I have to join in one of the program, which is because we have 34 institutions everywhere. Some programs are going on. So being a Vice Chancellor, I always used to be the part of inaugural function. But today I've seen that your talks is little interesting. So I would keep one device continuously connected with you so that I can hear you. So it's a pleasure that you have joined, and our all participant students have learned a lot from you. Your politeness, your humbleness, your contribution will definitely motivate our teacher, especially those who hear this program. And hope so that in future, as you are said that this is my email, you can raise the doubt. I try to give the answer. That humbleness normally at this age, people are not showing that, or they don't have time, or they may have any other issues. But hope so that you have shown that it's really pleasure for us, and hope so that in future also our association will continue. Thank you very much for joining this webinar, and especially to rather give good tips to our students. Thank you very much, sir.
Thank you so much. मैं वचन देता हूँ, मैं आऊँगा आपके पास
personally
one of these days when the things
this campus is such a beautiful, I can say that after seeing you can realize it is a big one, green one, lavish green, clean one. We have employed 300 people nearby villages. They are cleaning our campus, even road, cleaning of road morning, evening 2 times. So you won't find any dust, anything across this campus. That is what we are very environment savvy, and that's why we are, our all gardens are very beautiful. There are about 60 people.
I'll come one day.
Maintain the garden.
Thank you once again. Thank you.
Thank you so much. I would like to thank Provost sir also to be here with us. Thank you so much, sir. Thank you so much, sir.
Yes.
Thank you so much. I would take your permission to leave.
Yes, sir. Yes, sir. Definitely. I'm really very sorry.
Thank you. Not at all.
I have also to join in one another program of 12:30.
Yes, sir. I'm so sorry.
Thank you so much.
I want to see that once it is over, I can detach from here and join to that program. Thank you very much.
I'm leaving right now. You can join the other program now. नमस्कार नमस्कार
Thank
जय
you.
हिंद
Thank
जय हिंद
Hello.